We're talking about ways of tailoring antibody therapy using recently approved antibodies in multiple myeloma. I'm going to talk mostly about daratumumab and elotuzumab. I'm also going to talk a little bit about emerging antibodies with the same targets. So isatuximab and MOR202, as well as a conjugated antibody that targets SLAMF7. Well, I think what we're seeing already. We're seeing a lot of data at this meeting and at ASH last year, and published recently about combining these antibodies with standard therapies. The data that's out there now is in the relapse setting, and I think we're starting to see these move forward quickly. We're beginning to see some preliminary results from upfront studies. So we're combining drugs like elotuzumab and daratumumab with standard frontline regimens like RVD. And I think there's an expectation that those are going to have a really big impact on the efficacy of those regimens. So in my talk, I mentioned the fact that there are numerous ongoing trials utilizing each of these antibodies together with regimens like RVD, with Cyborg-D, with RevDex. And we're going to be getting readout on those studies in the next 1 to 2 years. I think it's going to have a really big impact. I think we're seeing both daratumumab and elotuzumab have a major impact on duration of response. Both of them seem to have that activity in higher risk patients that traditionally don't have the durable responses from chemotherapy. And we're seeing, particularly in the case of the anti-CD38 antibodies, we're seeing a deepening of response. And so they're not just longer responses, but there's more patients who actually reach an undetectable disease state, minimal residual disease negativity, which is right now really a holy grail in multiple myeloma. To cure patients, you have to render their disease undetectable, right? So getting a sizable number of patients to that state now is a major leap forward. So far the toxicity profile is really remarkably benign. Both the approved antibodies have infusion reactions associated with them. But to be honest, a lot of the toxicity we're seeing is the result of being able to be on therapy for longer, so just longer exposure to therapy. But it doesn't make the regimens dramatically more toxic. So it's really it's almost all upside. The conclusions of my talk is that we're entering an era where antibody therapy is going to be utilized in first or second line therapy for almost all patients. By incorporating them into those regimens, we're expecting longer and deeper responses, including patients who traditionally didn't benefit as much from chemotherapy. We're going to see more patients, more non-transplantable patients, get to those deeper responses than we've ever seen before. And the other thing we're going to see, which I touch on in the talk, is we're going to start seeing these antibodies used in multiple lines of therapy, most likely, with different partners than the ones that we've already studied in randomized studies. We're going to start utilizing these antibodies with pomalidomide and carfilzomib and other drugs as part of later line therapy as well. Right now there are several dozen trials with each antibody ongoing, and like I said, we're going to be starting to see readout from some of these studies in the next year and a half.